Whoops. Sorry, this is being so slow right now. Okay, there we go. All right, thank you everyone for being here today. So I'm going to begin my presentation with a short story. So back in 2019, I volunteered at a non-native species removal event in Southern California's Santa Ana River. I collected hundreds of non-native fish, but by the end of the day, I found 0 native species. So this experience really put into perspective how impactful and detrimental invasive species can be to ecosystems. So my name is Courtney Moulton. I am a biology major in my my last quarter at UC Riverside. And for my project, I studied the population cycles of one of the Santa Ana River's worst invasive species, the western mosquito fish, or Gambusia affinis. So mosquito fish are considered so detrimental as an invasive species because they've been introduced to so many ecosystems as a source of mosquito population control, but also because they reproduce in such great numbers. So the population cycles of mosquito fish have been well studied in their native ranges, which include the northeastern United States. And in these native ranges, mosquito fish use fat reserves as an overwintering strategy. So they store a lot of fat before winter comes and before temperatures drop and resource availability drops. Native populations also reproduce from February to August, and the offspring born earliest in the year have the highest fitness because offspring born later in the year do not have as much time to store as much fat before the winter. There is also an interesting trend in native populations, which I will refer to as the male annual life cycle throughout this presentation. So, unlike females, male mosquito fish cannot store fat after they have matured. So in native populations, males must delay maturation in order to store enough fat to survive the winter and reproduce the next year. And you can kind of see that trend in this graph, which is from a native population in New Jersey. So on the y-axis, we have percent of body fat. The line with larger dashes is mature males, and the line with shorter dashes is immature males. So in August, mature males have fat reserves that drop very low to about 5%. And in that same month of August, immature males have fat reserves that are almost 3 times as much as the mature males. So remember, mature males cannot store fat. And so when we see this increase in the mature male fat reserves from August to September, it is not because the August mature males are rebuilding their fat reserves, but instead, this spike in the mature male fat reserves indicates that the mature males that were present in August died, and they're replaced by the males that were immature in August that have enough fat reserves and that matured by September so that they can have enough fat to survive the winter and reproduce the next year. So this cycle repeats every year. And like I said, I will refer to this as the male annual life cycle throughout this presentation. So the population cycles are well studied in the native ranges of mosquito fish where there is strong seasonality and winters get very cold. But what is unclear is how mosquito fish that have been introduced to Southern California have adjusted their population cycles with the region's mild climate. So the question I asked is, how has the mosquito fish population in the Santa Ana River, which I call SAR throughout this, how this population has adjusted its population cycles with Southern California's mild climate? And with this question, I focused on 3 aspects of the mosquito fish population cycles: their fat cycling, their reproduction, and the male annual life cycle. So here I have my original hypotheses. So for their fat cycling, compared to native populations, I expected that the Santa Ana River population would have a reduced fat cycle. So they would store less fat in spring and summer, but also lose less fat in the winter. I also expected that the Santa Ana River population would have a longer reproductive season compared to native populations, and that they would have a reduced or no male annual life cycle would be present in the Santa Ana River. So I began this project by collecting about 100 mosquito fish from the Santa Ana River every month from November 2019 to October 2020. These fish were euthanized, preserved in formalin, and brought to the lab for processing. And in the lab, I began studying the fat cycling by conducting an ether treatment, where I weighed the fish before and after soaking them in ether. So ether is a chemical that draws the fat out of the fish. So weighing them before and after soaking them in ether allowed me to calculate how much fat each fish had stored. And you can see that process in this image on the right. They are soaking in ether, which was once clear, but it turns yellow as more fat is drawn out of the fish. Next, I studied their reproduction by dissecting the pregnant females and staging the development of their embryos by looking at their pigmentation, their eye spots, and their fin rays. The picture on the right shows some embryos that are in pretty early stages of development, and on the left, we have embryos that are very fully developed. And to study the male annual life cycle, I analyzed the male gonopodium through a microscope. So the gonopodium is the fin that males use to inject sperm into females. And as they go from immature juveniles to reproductive and mature adults, their gonopodium metamorphosizes over a 3-week period. So the picture on the left shows a female anal fin, but this is also what juvenile male anal fins look like before metamorphosizing. The picture in the middle shows a gonopodium that is undergoing metamorphosis, and you can see it's elongating. And the picture on the right shows a fully developed gonopodium of a mature male. So I classified all gonopodium, or all males with developed gonopodiums as mature, and all males with gonopodiums that were undergoing metamorphosis as immature. And for the data analysis, I used month as my main independent variable, and I saw how that impacted the proportion of fat of both males and females, the number of developing embryos, or the fecundity of mosquito fish, and also the proportion of immature and mature males throughout the year. So here are my results for the female fat storages. So on the y-axis, we have proportion of female fat reserves, and you can see a pretty cyclical trend throughout the year. This indicates that females allocate fat towards reproduction. So in the months when they are reproducing in the greatest numbers, those are the same months when their fat reserves are the lowest. Fat reserves then increase as females stop reproducing. And so, yeah, this shows that females use fat in order to reproduce. Also, this population has a peak in their fat reserves in November, whereas native populations have a peak in their fat reserves in September. So this indicates that the Santa Ana River population has delayed the time when they begin overwintering. So in conclusion, for the fat cycling, the Santa Ana River population begins to overwinter later in the year compared to native populations. And also, the Santa Ana River population stores more fat than native populations do. And so we can conclude that the fat cycling of the Santa Ana River population is different compared to native populations. So here are my results for the reproduction of the mosquito fish. So on the y-axis is the number of developing embryos. I did not include any unfertilized eggs in this graph. And you can see how high the fecundity is of the mosquito fish, or how much more embryos they are producing in May compared to all other months. And what's interesting is that this Santa Ana River population continues reproducing through August, whereas native populations stop reproducing by August. So from this, we can conclude that the reproductive season of the Santa Ana River population has extended a month later into the year. And lastly, for the male annual life cycle, here on the y-axis, I have the proportion of male fat storages. And again, we see a pretty cyclical trend throughout the year in their fat reserves. In pink are immature males, and in blue are mature males. So again, in July, the mature males have a very low drop in their fat reserves, whereas the immature males have much higher fat storages. So remember that mature males cannot store fat, and so the increase we see in mature male fat reserves from July to August does not mean that these mature males in July are rebuilding their fat reserves, but instead it indicates that these males that are mature in July die and they are replaced by the males that were immature in July and that undergo the maturation and become mature by August. Likewise, this graph shows the proportion of males throughout the year. So in blue is the proportion of mature males, and in pink is the proportion of immature males. And you can see in July, between July and August, there's a drop in the proportion of mature males. And again, this indicates that the mature males are experiencing a die-off between July and August, and that's likely because their fat reserves are so low and they cannot rebuild those fat reserves. So in conclusion, the male animal life cycle is present in the Santa Ana River population, and it is very similar to the male animal life cycle we saw in native populations. So just to summarize my results, for the fat cycling, we saw different fat cycling in the Santa Ana River population compared to native populations. We also saw that the Santa Ana River population has extended the reproductive season a month later into the year. And lastly, we saw that the male animal life cycle is very similar to that of native populations. So next steps for this project, it would be interesting to see what exactly has impacted the reproduction and the fat cycling of the population in the Santa Ana River, focusing on ecological conditions like temperature, water flow, or competition for resources with other species. However, I really hope that these results can be used to help aid in the conservation of the species that are native to the Santa Ana River, which include the Santa Ana sucker and the arroyo chub, which are both considered endangered by the IUCN Red Species List. So that's everything I have for my presentation. I'd like to give a special thanks to Dr. David Resnick, Samantha Lavelle, and Samuel Bedgood, who all mentored me throughout this project. I would also like to thank Derek Lee and Euridia Reynoso, who helped with data collections. And I would also like to thank the Chancellor's Research Fellowship for recognizing this work. And thank you all for watching.